Hello and welcome to this Technology Networks webinar, a novel perfusion technique for multiomic studies on hepatocyte aging. I'm Dr. Stephen Gibney, Science Writer for Technology Networks and your host for today's session. We have a fantastic presentation today featuring esteemed speakers Dr. Krisa Nikopoulou and Dr. Jürgen Eiberger. They will be sharing their expertise and experience in conducting multiomic studies on hepatocytes. Before we dive into the presentation, allow me to introduce our speakers. Krisa is a molecular biologist who received her PhD in molecular biology from the School of Medicine of the National and Kapodistrian University of Athens. She currently works as a postdoctoral scientist at the Department of Neurobiology of Aging at the Max Planck Institute for Biology of Aging in Cologne. Jürgen is a marketing and application specialist at Miltenyi Biotec. He received his PhD in biology from the University of Bonn. In his current position, he specializes in sample preparation and tissue dissociation, including the isolation of primary hepatocytes from mouse liver. Following their presentations, we'll have a short Q&A session. You'll be able to ask any questions you might have for our speakers. Feel free to submit your questions at any time during the presentations by entering them into the box on the right-hand side of your screen and clicking send. If you encounter any technical issues, please just click the chat box on the right-hand side of your screen and our team will do their best to assist you. Without further introduction, I want to hand over to our first speaker, Jürgen.
Hello and welcome to our today's webinar where Krisa Nikopoulou will present her findings on her in-depth multiomics analysis of functional and healthy hepatocytes to understand the effects of aging. My name is Jürgen Eiberger and I'm Marketing and Application Specialist at Miltenyi Biotec. Krisa obtained the hepatocytes for her research with our new and automated perfusion technology. The simplification and standardization of the perfusion procedure could also be of interest to you. So that's why I would like to briefly introduce this new perfusion technology that Krisa used for her research on the next few slides. Before I start with the presentation, please note this disclaimer. So isolating primary hepatocytes from liver tissue is a very complex and challenging procedure. If you do it conventionally, you usually work with anesthetized animals, and that means you always require an official animal experimentation approval by the different authorities. For this reason, often only a few people in the lab are allowed and able to do the manual perfusion. And this is how the manual perfusion setup normally looks like, with a water bath, the peristaltic pump and the needle, for the cannulation. And after anesthetizing the animal, you open the abdomen and then you have to cannulate the vena cava with a needle. This is a big challenge because you have to hit the vena cava perfectly. Then you use buffers and collagenase to perfuse the liver, and you have to adjust the speed of the peristaltic pump. And when the perfusion is done, you take out the liver and dissociate it manually to obtain a single-cell suspension, which contains mainly primary hepatocytes. So this procedure requires very experienced and trained people. I would like to show you now how we simplify perfusion with the gentleMACS technology. It's an easy, automated, and operator-independent procedure that is controlled by the instrument you see on the right. That's the gentleMACS octodissociator with heaters. The procedure is highly reproducible and standardized. Everyone can use it, and it's very easy and fast to learn. So we work with resected organs from sacrificed animals, so no anesthetization or manipulation of anesthetized animals. We do the perfusion with only one liver lobe. When we talk about mouse liver perfusion, we take the biggest liver lobe, the left lateral lobe, and with rat, we usually take a smaller one, the right dorsal lobe. So you don't need to do any cannulation of blood vessels. And because we also use our own enzymes and reagents, which have a high lot-to-lot consistency, the perfusion is highly reproducible. The gentleMACS perfusion technology is an easy-to-use and automated system. The required equipment you need to perform this in your lab is the gentleMACS octodissociator with heaters. It's the device that automates the process and guides the user through the workflow. The gentleMACS perfusers are single-use tubes in which the perfusion is performed. The liver perfusion kit provides the required buffers and enzymes. and the gentleMACS perfusion sleeves are required to drive the perfusers. These are the link between the instrument and the perfusion tube. The very special thing of the technology is the gentleMACS perfuser. Let's take a closer look at it. On the left side, a photo of the perfuser. On the right, the individual parts. The perfusion is performed inside the perfuser. The perfuser contains the needles. And on the downside of the tube, there is a membrane that serves as a peristaltic pump. So after tissue resection, the tissue is placed on the grid, and with the clamp, the tissue gets fixed. The whole upper part with the lid is then connected to the clamp, and finally everything is placed in the base, and during the assembly the needles in the base pierce the tissue, and now everything is prepared to proceed with the perfusion on the gentleMACS instrument. So what drives the pump and how does the perfusion work? So on the right you see a schematic drawing of the inside of the tube. The tube is assembled and filled with buffer. You see the tissue in red, placed on the grid and pierced by the needles. These are the needles shown in blue. The lobe is pierced randomly by the 4 needles, so you don't need to hit a vein, but it is important that we have a liver lobe with an intact capsule. The needles are placed in the center of the lobe with a capsule around it, and now we can float the tissue with the different buffers while the capsule keeps the liquid inside the lobe. In yellow, the membrane on the downside of the tube, which serves as a peristaltic pump. And the gray thing here, down here, is the sleeve that rotates and drives the pump. So how does it work? The buffer inside the tube enters the membrane, which serves as a peristaltic pump, via this opening here. The sleeve down here drives the pump by rotation, and the liquid is pumped through the needles into the tissue, and the tissue gets perfused. Let's have a quick look on the timeline. The perfusion workflow with our technology takes about 45 minutes. It starts with the removal of the liver from the animal and the assembly of the perfuser. The perfuser with the sample is then placed on the gentleMACS, and the instrument guides the user through the individual steps, during which the buffer needs to be exchanged a few times. The perfusion is complete after 25 minutes, and the tissue is pre-digested by the enzymes. The subsequent dissociation of the tissue and the release of the hepatocyte is done using another tube, the C tube, which is designed to dissociate the tissue and release the hepatocytes in suspension. And this is the C tube, and here's the perfuser. This takes further 5 minutes, and it is also done using the gentleMACS instrument, and it results in a very nice single cell suspension. Finally, the hepatocytes are collected with a short spin. NPCs remain in the supernatant while the hepatocytes form a pellet. That way, we end up with around 10 million hepatocytes. Since we only simplify the perfusion method, the hepatocytes can be used for the same applications as you do after manual perfusion. Summarizing the advantages of this technology compared to manual perfusion, so it's easy to use, it's automated and operator independent, so it's highly reproducible no matter who does it, and everyone can use it without extensive training. There is no need to do the perfusion on a live and anesthetized animal. Livers from sacrificed animals are processed. This means less animal suffering, and it also means less effort when applying for animal experimentation proposals. Since perfusion is done with one liver lobe, remaining liver and organs can be used for other applications, and you can perform up to 8 perfusions in parallel on one instrument. There is no loss of animals due to unsuccessful perfusion. So if you are interested in this technology and you would like to test it, please feel free to get in touch with us or simply leave us a message in the chat. So if you would like to get more detailed information on the gentleMACS perfusion technology, you can also watch this recorded webinar shown on this slide entitled Automating Liver Perfusion to Isolate Primary Hepatocytes, which was presented by Philipp Schlermann last year. So Philipp is the global product manager of this technology, and in his webinar, he explained even more detailed the gentleMACS perfusion technology. As you can see, Philipp's webinar is also available via the Technology Networks platform, and it can easily be found using the search function, or you scan the QR code and watch it on demand whenever you like. And now we are looking forward to Krisa's presentation. So please, Krisa, I hand over to you.
Okay, hello everyone, and thank you for Jürgen, and thank you for the introduction. It is my pleasure to share with you today our work on spatial and single cell profile of the aging mouse liver. The data that I will discuss today with you were recently published, but in this presentation I will focus more on the technical aspects of our research, and more specifically how we have set up our experiments to study the epigenetic, transcriptional, and metabolic profile of hepatocytes derived from young and old mice. During aging, there is a general physiological decline in metabolic functions, often connected with gene expression and epigenetic changes on the organismal, on tissue-specific, and also on the cellular level. The liver tissue is a key metabolic organ that is comprised by almost 80% of hepatocytes. The rest are non-parenchymal cells like endothelial cells, Kupffer cells, other immune cell types, etc. Throughout the liver, the blood flow from the portal vein and hepatic arteries to the central vein of the tissue creates a gradient of molecules and nutrients, establishing a division of metabolic labor to hepatocytes. So hepatocytes, depending on their proximity to the portal or the central vein, although they are histologically similar, exert different metabolic functions, a phenomenon that is also known as liver metabolic zonation. We were intrigued to understand how these metabolic profiles are connected to the epigenetic profiles of the cells and how the zonation of the hepatocytes affects their aging phenotype. We have set out a study of omic approaches in combination with functional assays and histology using livers from young, 3 to 4 months old, and aged 18 to 22 months old mice. Initially with classical histology H&E staining, where the nuclei are visualized in purple and extracellular matrix and cytoplasmic structures are in magenta, we have observed an increased deposition of fat, these white unstayed areas that you can see here in the old livers. We have confirmed with oiled red O staining, which stains the red and neutral triglycerides and lipid droplets on frozen tissue sections, that these structures are indeed lipid droplets. So we wondered if this increased amount of lipids is related with a disease phenotype. Therefore, we have used Sirius red staining. Sirius red stains with red the collagen fibers that should increase in fibrotic tissues. And as you can appreciate from the images here, there is no apparent fibrosis in the old liver. So the old livers that we have used, despite the accumulation of lipids, they seem to be relatively healthy. We turn to lipidomics in order to understand what is the lipid constitution of these old but healthy livers. We have isolated lipids from old and young livers and observed a general lipid remodeling during aging, shown here with this PCA plot with a rewiring of several classes of lipids, like an increase of specific classes of triglycerides and diglycerides, diacylglycerides, or changes in membrane lipids that you can see in this heat map here. Interestingly, we have also observed a decrease in ubiquinones. These are the coenzymes Q9 and Q10. These lipids are mitochondrial electron transport chain lipids, and we wondered if the reductions in their levels indicates changes in the mitochondrial metabolic function with aging. Since metabolic functions in the liver, as I mentioned before, are zonated, we wanted to understand what regulates their changes during aging on the transcriptional level. For this reason, we have decided to perform spatial transcriptomics. Spatial transcriptomics is an approach that can give you high throughput information about the spatial distribution of mRNA molecules in the different zones. We have chosen 2 marker genes CYP2F2, and GLUL that are expressed in higher levels in the periportal and pericentral area of the tissue, respectively. And we have used these genes to define the zones on our spatial transcriptomic datasets. So we used these genes to later assign the expression of other genes as periportal or pericentral. Therefore, every gene that is expressed within the GLUL areas is considered to be pericentral, and every gene that is expressed within the CYP2F2 areas is considered to be periportal. We have confirmed the specific localization of the expression of our marker genes using RNA-scope, which is an in situ hybridization method to detect RNA transcripts on tissue sections. From this experiment, it is quite obvious that the expression of GLUL, here shown in magenta, is localized at the central vein of the tissues, and the expression of CYP2F2 here in blue at the periportal areas. And you can also appreciate that the expression patterns of these two genes are mutually exclusive. A very interesting observation that we had with these experiments is that the localization of lipid droplets is very localized in the old liver tissues around the central veins. So one of the most exciting results of our spatial transcriptomics were the pathways of genes differentially expressed during aging. In the pericentral area, these genes are connected to lipid metabolism, a result which correlates well with the RNA-scope, showing that large lipid droplets accumulate in the pericentral area of old livers. Whereas in the periportal area, we see changes of genes associated with the mitochondrial electron transport chain. Since we have already identified with lipidomics a reduction on the levels of ubiquinons in the old livers, we wanted to understand if these changes in gene expression and lipid constitution somehow affect the function of hepatocytes in the two different zones. So to perform these functional assays, we needed to isolate available hepatocytes from the portal or central area. Luckily, at the time we were planning those experiments, Shalev Itzkovic's lab have just published a study showing that pericentral and periportal hepatocytes express surface markers that can be used to separate the cells from the different metabolic zones with flow cytometry. These markers are CD73, shown here in red and expressed from hepatocytes around the central vein, and ecadherin, shown here in green, and expressed in the periportal area of the tissue. Therefore, in order to be able to isolate periportal and pericentral hepatocytes, we had to perfuse young adult mice, isolate the hepatocytes, and stain them with CD73 and ecadherin, and separate them with flow cytometry. We then did qPCR to check the expression level of the marker genes mentioned before. These are GLUL and CYP2F2 to confirm their expression in the pericentral and periportal hepatocytes respectively. And we seeded the cells in order to perform a metabolic sequence assay using the mitochondrial stress kit to assess the metabolic function of the mitochondria. I'm going to explain a little bit more in detail how technically we achieved this experiment, because this was the first time and that we have extensively utilized the perfusion kit from Miltenyi that Jünger mentioned before. This proved to be an asset for the progress of our experiments. Initially, we started perfusing the animals through the vena cava, which is technically challenging for those that are not experienced in the perfusion procedure, like us. And in addition, this technique created an extra burden for the animals, as they had to be anesthetized first and then perfused while they are under anesthesia. By using the perfusion technology from Miltenyi, we just needed to isolate an intact lobe from the liver tissue and after dissecting the animals. And this lobe produced 10 to 20 million viable hepatocytes on our hands, depending on the age of the animal and the size of the lobe. The whole procedure was very fast and very reproducible, something we needed in our experiments for mainly two reasons. First, the number of old animals was always limited, since very often the black 6M male mice that we used in our experiments developed liver tumors at old age. So we couldn't use these mice, and we wanted 100% success perfusing the healthy mice that we had in our hands. Second, the levels of ecadherin, as you can see here, are lower in old hepatocytes. so the final number of cells available for experiments after flow cytometry was often very limited. Therefore, getting easily a big amount of valuable hepatocytes was indispensable for the progress of our sequence experiments. After seeding the pericentral and periportal hepatocytes and performing the sequence metabolic assay, we can see that although with aging the basal respiration and ATP-linked respiration remain unchanged between the zones, the spare respiratory capacity and maximal respiration, which are indicative of mitochondrial health, are greatly reduced in all periportal hepatocytes. This result correlates well with the age reduction of ubiquinose that we have observed with lipidomics, as well as the mitochondrial electron transport chain pathway genes that seem to be differentially expressed in the old periportal hepatocytes that we have seen with our spatial transcriptomic data. To sum up so far, we have seen bigger lipid droplets accumulating around central veins and reduced mitochondrial health in periportal hepatocytes. Our next question was, how are these phenomena regulated, and is the epigenome involved in the aging process of the different zones? We decided to perform single cell attack, which is a technique that can identify the accessible chromatin in individual cells. This method is ideal to assess the different cell types of the liver tissue. As you can see in this UMAP, the liver cell types indicated here with different colors are clustering with each other, and we can discriminate most of the major liver cell types like the hepatocytes, endothelial cells, Kupffer cells, Ito cells, and progenitor and immune cells. We have additionally color-coded this UMAP, separating the young from the old cells, and we can clearly see that hepatocytes, which are this big cluster here, show the most profound separation of cells with aging, meaning that the epigenome is very different between young and old hepatocytes. So we have used our single cell attack data to see how chromatin accessibility can predict the aging phenotypes that we have identified so far. One striking example is the CDA gene locus. The CD family of genes have been associated with the formation and accumulation of large lipid droplets, a phenomenon that we have already observed in our aging livers. By using our single cell attack data, we can predict the co-accessible size of one locus. In this case, for the CDA gene promoter, we see a large number of co-accessibility at the gene and upstream of the promoter, which correlates with increased deposition of H3K27 acetylation, a marker for active enhancers, indicating that the gene is very prone to be expressed. Indeed, the spatial transcriptomics data show that the CDA gene expression is increased in old livers, especially in the pericentral regions, where we have also observed the accumulation of large lipid droplets. Additionally, we performed electron microscopy in order to confirm the large lipid droplets with aging. And as you can appreciate from the images and the quantification plot shown here, the old hepatocytes have significantly larger number of lipid droplets with age. Finally, we wondered if chromatin accessibility correlates in general with changes in gene expression in individual cells during aging. So in general, the question is how the epigenome regulates gene expression. And we focused on hepatocytes because those show the most profound aging differences so far in all of our experiments. By analyzing publicly available single-cell RNA sequencing data, we have seen that the correlations were not always straightforward, as in the case of the CDA gene that I mentioned before. And we have also seen that the gene expression becomes very variable in old hepatocytes. In order to understand if the zonation of hepatocytes, so the proximity to the central or the portal vein, accounts for this variability and what genes contribute to the variability in gene expression with aging, we have decided to perform single-cell RNA sequencing with the SMART-seq3 technology, which can identify more and even lowly expressed transcripts, giving a more complete picture of the transcriptome of single cells. Here, again, we perfused mice, young and old, using the Miltenyi liver perfusion kit, and we isolated hepatocytes and separated them with flow cytometry according to their ploidic profile. So we stained the hepatocytes with host. We did this to account for any difference that may occur due to the different ploidy of the cells. And by using the coefficient of variation, we can see an increased variability in old hepatocytes, which is even more profound in the diploid old hepatocytes and in the pericentral old hepatocytes. So gene expression with aging seems to be resonated and variability seems to be resonated and ploidy dependent. What I'm not showing to you here, and is a very interesting result that you can also find in our manuscript, is that the genes that are differentially expressed between young and old hepatocytes are standard genes changing with age and have to do with xenobiotic, drug metabolism, lipid metabolism, and mitochondrial function. Whereas the most variable genes that change with aging have to do with regulation of transcription, splicing, and gene expression. And this result confirms the notion that the epigenome and transcription do not correlate with age because the levels of gene regulation are somehow disconnected in the aging process. So which molecular mechanisms play a role in this regulation or dysregulation of transcription? We don't know yet, and this remains an open question. With that, I want to summarize the bottom line of our results. Location of the hepatocytes in the liver affects their aging profiles and phenotypes on the functional level or on the level of the epigenetic regulation. And finally, I want to give credits and thanks to the people that we work together on this project. Of course, my lab, Peter Tesserts, Swati and Nick, my collaborators, the facilities in the Cologne campus, and also Miltenyi that offer the opportunity to test their perfusion technology and made our research simpler and easier. I'm looking forward to take your questions. Thank you.
Thank you, Jürgen and Krisa, for those presentations, and thank you to the audience for listening. I hope everyone found that presentation as insightful as I did. We're now going to move on to the Q&A session. You will have the opportunity to ask your questions to Jürgen and Krisa. To do so, use the chat box on the right-hand side of your screen. Type in the questions and click send. We've got about 15 to 20 minutes for questions today, so we'll try and answer as many questions as we can in the time that we've got left. We've already had a few questions come in, so I think we'll just dive straight into them. This one, I think, is directed more towards Jürgen, so we'll start with you, Jürgen. How difficult is it to properly place the liver lobe in the perfuser?
Yes, thank you. Our perfusion technology is very simple and easy to learn. So you only need to know how to resect the liver lobe and how to assemble the tube. And maybe one thing everything should be done quickly, that the blood does not start to coagulate. So it should not take more than 5 to 10 minutes from resection to the start of the perfusion. But that's it. So everyone can learn it quickly, and everyone in the lab will be able to do it. But I don't know, Krisa did this already in the lab. So Krisa, was it difficult to set up the new perfusion technology in your lab?
Actually, not really. Of course, we had the training for the first time how to use the kit, which lasted 2-3 hours maximum, and how to use the whole machine for dissociation. But in the beginning, that we were not very experienced, we were not handling more than 2 tissues in parallel, because really we needed to be fast. So I think after a couple of times, or maybe 3 or 4 trials, we started perfusing 4 to 6 tissues in parallel, which was doable within 7 minutes of time. So it was really easy afterwards. Yes.
Fantastic. I think you've addressed some of the questions that we've already had about kind of the difficulty of implementing it. So the other question we've got, which is along a similar line, so we'll kind of start with you, Jürgen, and then Krisa, you can kind of comment. So what would you say the training period is to feel confident when running the liver perfusion technology?
I think that's it doesn't take long. So if you do it once or twice, and then you are familiar with this technique, so as said, so you only have to assemble the tube and know how to do it. And yeah, so there is no long training needed, so everyone will be able to do it.
I would also say that the protocol that comes with the kit is very self-explanatory. Of course, for us, it was the privilege to be like the beta testers of the kit. So we had someone really showing to us how we can assemble the whole perfusion machinery, let's say. But the training lasted around 2 hours, and then we repeated the procedure the next 1 hour. And basically, afterwards, I trained a couple of people in the lab that were working with me. We did it one time together, and everyone was independent afterwards. So it was really easy to implement it in our lab, at least.
That's good to hear. I think people will always enjoy hearing that something like that is quite easy to set up and get going. So, Chris, I think we've got more of a technical one here for you. Can you elaborate on how mRNA splicing and mRNA stability could be affected in aging hepatocytes? Will this be something you're looking into in the future?
This is a really nice question. So, regarding splicing, of course, we know from the literature that alternative splicing is really deregulated in aging, and there is a study that shows that 40% of the genes in the human mouse brain are affected during aging through alternative splicing phenotype. And there are also studies that show that interventions that promote longevity, like caloric restriction, dietary restriction, are also affecting splicing events. So splicing is a process that is changing during aging, and it could explain, at least in part, the disconnection that we see between transcription regulation and chromatin. We haven't looked into that into our manuscript, and I think that could be an interesting aspect. Although in a parallel study that we had done in the lab, there was more a connection to mRNA stability. And I would say not mRNA stability per se, but the regulation of mRNA stability. So basically, we see with aging the steady state levels of RNA, they don't change. And also, nascent transcription remains largely unchanged. But what we see changing is the chromatin accessibility, that it becomes more increased, and we see also a decreased posing of the polymerase on the promoters of several genes. And this decreased posing could be due to decreased stability of the posing complex, which could lead to premature termination of transcription. Thus, it could lead to destabilized mRNA. But in order to really look into these processes, you need other approaches, like SLAM sequencing, for example, where the mRNA would be labeled with 5-thiouridine, and then synthesis and degradation can be checked. So I think that would be a plan in order to check stability and in order to understand why there is this disconnection between how the accessible chromatin regulates transcription. Of course, we haven't done that yet. There are publications that they look into mRNA stability in aging, but do not really go into much detail if the stability is changing or the degradation is changing. So this field is yet very unclear. I don't know if this answers the question, hopefully.
Yes, I think it does. It did a great job.
Okay.
Yeah, thank you very much. We've had a few more questions. Again, Chris, another more technical one. How do the variable levels of ploidy in hepatocytes influence your observations regarding gene expression or chromatin accessibility?
So for the chromatin accessibility for the single cell attack data, we have normalized our data sets according to coverage, because we know that with aging, ploidy is increasing. So we wanted to be sure that we don't have increased reads in our data set because of increased ploidy. In the mRNA, we accounted for these changes because we really we really sorted for diploid and tetraploid hepatocytes. And in our study, we see that there is an increased variability in the diploid hepatocytes. And in a study that was published very recently from Celia Martinez, I think they have also used the perfusion technology in their paper. So they see that the increased ploidy of tetraploid hepatocytes in aging somehow buffers the aging phenotypes. And maybe this already explains why we see increased variability in the 2N hepatocytes compared to the 4N hepatocytes in aging. So I think that ploidy is a factor that we should take into account when we look into hepatocyte aging. And we briefly touched on this matter, but of course, we didn't normalize all our data according to ploidy. This was not possible.
Fantastic. Again, carrying on with you, Chris, I think we will switch back to Jürgen, but we've had one here about data sets. So is there any indication in your data sets that the old livers are more prone to disease progression? Have you checked any marker genes that could potentially be linked to disease phenotypes?
So that's an interesting question. Thank you very much. We got it also during the revisions of our paper. Actually, we didn't check for disease markers. We initially, as I showed, we checked if there is any fibrosis, apparent fibrosis in the tissues. And since we see increased lipid accumulation, we were expecting that the tissues would be at least fibrotic. And that would explain that the mice have non-alcoholic fatty liver disease, for instance. But our mice were relatively healthy. And whatever tissue had increased fibrosis, we didn't use in our experiments. The lipidomic data can be of interest if someone wants to check disease progression. We didn't know how to do that. There are not a lot of lipidomic data sets available for liver so far. So maybe there are some. I think there is one study that shows that phospholipids increase in the percentile area in some diseases, but in our data, we haven't seen any clear signature. We only checked, as far as I remember, for aging markers, let's say, and these were the P21 senescence marker, which was increased in our old livers. We have also seen decreased mitochondria, decreased the length of telomeres, but nothing else. So these are markers of aging mostly, but not disease actually.
Fantastic. Thank you again for that. Just a reminder to the audience to keep dropping your questions in the Q&A box on the right-hand side. We have had quite a few. We'll probably take another 5 or 10 more minutes to ask the questions we've got, but keep dropping them in the chat. So I think this one is a bit of a broader scope about the technology. So I think this one might go to you, Jürgen. There's kind of 2 in 1 here. So is it possible to perfuse human liver with this technology? And also, are there efforts to develop the technology for tissues other than liver?
Yes, thank you. That's indeed a most frequently asked question. So unfortunately, processing human liver samples is one of the limitations we have. So of course, the liver sample has to fit into the tube, but even when you have only a small tissue piece that is small enough to fit into the tube, human liver is challenging, but this is this applies to manual perfusion as well, so it's always challenging. And The challenge in perfusing the human liver with our technology is that we need to have an intact capsule, which we don't have when we have liver biopsies. So we know that there is a high demand on perfusion of human liver and human hepatocytes. That's why we are working on that. But so we hope that we are able to perfuse the human liver samples in the future. However, what already works is the perfusion of humanized liver. So here we do have this intact capsule we need. So perfusion of human liver is not yet possible, but we hope to find a way to make it possible in the future. And regarding the fibrotic liver, so it is possible to process fibrotic liver as well. So also other diseased livers, MASH liver or fatty liver. As long as you have an intact capsule, it is possible to use our technology. But have in mind, so if you have a challenging sample and the yield is lower with the manual perfusion, it will be probably also lower with us. So to answer this part of the question, so yes, perfusion of fibrotic liver is possible, but the yield might be reduced.
Fantastic, thanks for that. So then we've had another one here. How much workup has been done on the downstream isolation of NPCs using this approach? Are there any changes in endothelial metabolism with aging in these mice too? So I think, again, Chrissa, that one's more for you, possibly.
Okay. Now maybe I can take the first part. So
yes, the first part, yes, because I haven't.
So it's right. So we can get both hepatocytes and the macrophages and endothelial cells and other NPCs. So so this is, let me think about the answer a little bit. So far we talked about perfusion. So, but perfusion is not necessary to get the NPCs. So when you only want to obtain macrophages and endothelial cells, perfusion is only necessary when you want to get the hepatocytes. With the perfusion technology, you get both. But to obtain macrophages and endothelial cells, you don't need to perfuse the liver. You can use another workflow we provide, also with the gentleMACS. That's just the dissociation of the liver tissue. And this makes it even more simple. So, and you can process even more tissue. So with the perfusion, we only use the left lateral lobe. And for the dissociation, you can use more starting material, and that's why you will obtain more NPCs in the end when you follow this workflow. So you have two options. You can perform a perfusion and obtain all the cells, hepatocytes, macrophages, endothelial cells. So you will get all cells from one sample, or the second option is you just perform the dissociation on the gentleMACS and obtain macrophages and endothelial cells, but you won't get any hepatocytes. And for the functional analysis of the NPCs, I would like to hand over to Chrissa, so we didn't do this.
Yes, I want to also add that I have tried also the dissociation of liver kit, not for this project, for a completely different project, because I needed cholangiocytes. Really I had a very good yield of cholangiocytes for the other project that I wanted to create, liver organoids. I haven't isolated any NPCs after the perfusion, although it is possible, as Jürgen said, because after the perfusion, I think it was shown in both presentations, we use debris removal solution, which creates a gradient, and actually in one part of the gradient are all these small cell types, but we have never worked with these cell types. So this is, again, a question that I have recently gotten when I discussed this data. what happens to the non-parenchymal cells, and why don't you see some, or why don't you comment on any changes with aging? So I think that one part is our epigenetic profile. The technology that we are used is single cell attack. So the epigenetic profile that we got from the non-parenchymal cells did not show any clear aging phenotypes. We had cells both from young and old mice, as you have seen in the UMAPs, but there was no clear separation in the UMAPs between young and old. On the transcriptional level, we really analyzed several spots of the spatial transcriptomic data. So if you remember, the spatial transcriptomics do not give a single cell resolution, but actually single spot resolution. That means that in every spot there are several cell types. Of course, the majority would be hepatocytes because this is the more abundant cell type, but other cells are also there. And we analyzed some markers for these cell types. So for Kupfer cells and stellate cells, and also endothelial cells, we have also some markers in the manuscript analyzed that we see differentially changing with aging. And these markers are connected to inflammation and senescence. And I think it's very important to look in the non-parenchymal cell types, because since they are changing with aging, and this is known also from other publications, they would really affect the microenvironment and the metabolic profile of the hepatocytes per se. In our study, we haven't done any analysis at all for the non-parenchymal cells. So I think that this is a part that is missing, and maybe the metabolic changes that we see in the hepatocytes are highly attributed also to the neighboring cells, of course. So I would comment that that's an interesting follow-up.
Fantastic. Thank you very much for that. We've had quite a while, I think, one here for you, Chrissa. The effect of spatial distribution in functionality and fate of the cells within tissues is becoming obviously more evident. They do complement your work here, so it's saying that you showed some very nice examples in the liver. Do you picture that the specific tissue architecture can drive specific aging trajectories within potentially other organs? And which other organs would you think is likely to share similar age-related changes in transcriptomic and metabolomic reorganization?
So I am not an expert in many organ cell types, but I would say that every organ that is that there is blood flow, of course there is a zonation. I have read a paper about kidney where there is a zonation of immune cells, for example, created by the blood flow. So I would think that this zonation of immune cells may affect the other cell types also in the kidney, for example, the podocytes or other endothelial cells. And according to their location to the kidney, if they're close to the urinary tract or not, maybe they're more susceptible to infections. This could be an interesting study for the aging kidney, for example. I'm not aware if there is a study at all regarding the epithelial cells of the kidney, but there is an architecture in this organ. And of course, now I started working in a lab that is focused on the brain, and the brain has a very detailed architecture with different brain regions, and every brain region is populated by different kind of neurons. And also within the same brain region, neurons can have different, like, aging trajectories or different epigenetic profiles. So, but I wouldn't be very narrow-minded only about kidney or brain, or maybe the heart if it has a specific architecture. Maybe every organ that there is blood flow, there is changes that happening in different locations, I would say. Yeah.
Fantastic. Thank you very much for that. I think that is all the questions that we've had come in. We might have a few more, but I think we will start to wrap up now. Thank you very much, Chris and Jürgen, for both your presentations and also for obviously taking the time to answer the questions today. Any questions that do come in that we couldn't address during the session, we will answer them offline as soon as possible. You can continue to ask questions when watching the webinar on demand. We'll send them off over to the speakers offline. Finally, don't forget to download your certificate of attendance from the handouts tab on the right-hand side of the webinar platform. Once again, thank you very much, Chris and Jürgen, for your time today, and thank you to the audience for listening.